Hi all. Today we'll discuss a model describing a spread and maintenance of epigenetic information. We'll call it painters in chromatin. So here on the left, we have a high resolution image of the nucleosome, one of the first. We know that we see that it's a very complex structure, but for the discussion today, we'll have a form of a spherical curve assumption of the nucleosome. So epigenetic marks regulate chromatin state, and we can have a host of different epigenetic marks on histones. And these marks are actually deposited, or these modifications are brought in by histone modifying enzymes. And the machinery, or the molecular machinery that brings in these modifications, are quite diverse and complex and vary from species to species. So the question which we are asking today is, or trying to address, is like, can we have some generic or unified principle for this particular process? Like, can we zoom out and sort of have an all-encompassing picture which would describe this process in different species? So the three key elements which we think we are which would help us in doing that is one, the sequence-specific recruitment of these enzymes. So here we assume that the modifying enzymes bind to some DNA binding proteins and have some specific affinity to the sequence itself. The other one would be reader-writer ability of these enzymes. So wherein these enzymes have a reader domain, it reads the mark on one nucleosome and writes the same mark on adjacent nucleosome in 3D. And the spreading process, like I said, happens in 3D. So it's not just in cis. Long range propagation can happen via chromatin looping. So at a TAT scale, it can be between enhancer promoter and it can be due to enhancer promoter interactions. So today we'll try to understand how these mechanisms contribute to spread and maintenance of epigenetic marks. And finally, we'll relate it with some of the recent experimental findings. The painter model. So here, the nucleosomes can exist in two states, modified M and unmodified U. So modified having some modification which affects local chromatin state, and unmodified having no modifications which affect chromatin state, so the native nucleosome. So we are actually lumping all the modification and complexity of the nucleosome into a two-state situation here. And the transition between these are modulated by the transition propensities RMU and RUM. So here we'll focus on the spread of modified state M. And in order to do that, like in the transition propensity RUM, that is the one that takes unmodified state to modified state. In this particular term, we will lump in all the mechanisms which we discussed before, the sequence-specific recruitment, reader-writer ability, and long-range spreading via chromatin looping. So this is my DNA sequence here, like with blue and yellow nucleosomes. And this is a specific site where I have sequence-specific recruitment. So here, there are enzymes which are bound to the DNA sequence, which bring about, so these are histone modifying enzymes, which cause the spread of modified state M. And as you see here, like these enzymes can actually be recruited by modified state itself. Like, so the nucleosome in modified state can recruit these enzymes as well. So this is a scheme of our spreading process. So the process of sequence-specific recruitment can be mathematically written down like this. So here we have the writing rate of the enzyme. Rho Wj would basically give us the probability to find the writer or painter at position J. And PCIJ gives us the probability to find nucleosome I co-localized with nucleosome J. So that basically takes care of the chromatin looping or propagation in long range. And since we would like to keep it simple and we are specifically focusing on the spread of modified state M, we keep the reverse process, the process that takes modified state to unmodified, like we keep it simple, and we lump all the processes like enzymatic removal of the marks, uh, replication, and nucleosome turnover into a constant term K0. So let's look at a 200 nucleosome region like this. And so we start off initially with random state, like random blue and yellow nucleosomes, like we see here. And at position 0, we have the region where sequence-specific recruitment happens. So we have enzyme recruitment here. And then we let the system evolve in time, where the chromatin state dynamics is modulated by these propensities. So what we see is, like, we see a clustering of blue states around the sequence-specific recruitment site. Uh, to illustrate this process, I have an animation here. So these are our enzymes or painters. So they're recruited onto the specific sequence here. And we see a clustering of blue states around that sequence-specific recruitment in time. Now, the other key element of our spreading process, the reader-writer term, how does that affect? So here, what happens is that, like, the modified state can actually recruit these enzymes. So it's no longer just bound to the sequence. We have enzymes which are recruited by the nucleosome in blue state or modified state, and then it affects the spread around the 3D region, around the 3D vicinity of the particular recruitment region. And if we compare it between the previous states, so we have the blue states are a bit more spread out from the painter or the sequence specific recruitment state. So to illustrate it better, we can look at the probability to be modified. Uh, so earlier we were basically looking at a single cell level. So now we'll actually be looking at an ensemble of cells, so an average quantity. So this is an observable, like, probability to be modified, can be thought of analogous to some ChIP-seq profile. So case one would be the green curve, like where we don't have any any reader writer term. So there's only sequence specific recruitment here. So we see clustering of blue or modified state around the recruitment site. So here the recruitment region is marked in yellow. And when I switch on the reader writer term, so I increased delta. So delta can actually be thought of as some binding affinity of the enzyme to the blue state or nucleosomes with modifications, or nucleosomes in modified state. So I switch on delta, and then I see, or what we see is, like, some characteristic heavy tails. So if I increase delta further, we see heavy tails. This means that the blue state or the modifications are much more spread out and not just localized around the painter region. So by looking at the profile, so the shape of the profile is characteristically different when we incorporate different mechanisms. So by looking at the profile, like, we can actually comment on the underlying process. Like, so if you look at the slope here, which sort of goes like 1 by x, so it sort of indicates that we just have the sequence specific process, and when it is different and when we start to see heavy tails, we expect some reader writer coefficient as well. Now we would like to relate this to some experiments and see if we can make experimental predictions. Here on the left, it's in mouse embryonic stem cells, we are looking at H3 canine ME3 marks, and we can actually predict the canine ME3 profile around a transposon element with the sequence-specific painter model. On the right, what we have is a phosphorylation profile in human cells. So here we have a double strand break at position 0. And this is the profile of the enzyme. Like, this is the kinase that maintains the genome integrity. So it is very localized, highly localized at the break. And then we have phosphorylation profile, something like this. Like, so this is an average of over 80 cells. So here we can see that there is no reader-writer process because the enzyme is highly localized and the only recruitment is seen only at the double strand break. So this is a classic example of the sequence-specific process. And we can very well predict this phosphorylation profile using our painter model. So we know that these enzymes have residence time, like they're not bound to the DNA forever. So they unbind after a specific time. So now we look at how the region behaves, like how the marks evolve when the sequence-specific enzyme unbinds. So at t equals 0, what we have is some profile established by the painter model, like which exactly like the one which we have seen. So we have some profile at time t equals 0, and then at t equals 0, the sequence-specific enzyme unbinds. And then we ask the question if the reader-writer process is able to maintain the states or not. So when delta equals 0, like no reader-writer process, we see exponential decay of the modified state or modifications. It quickly turns into unmodified state here. And with low reader-writer process, we see the same. And at a critical reader-writer process, we see a slow decay of states, like very slow. And above that threshold, we are able to maintain the states. So with sufficient reader-writer term, we can actually maintain the states. And the question is like, is the state at time t greater than t0, like the state which is maintained, is it related to the state that was at the beginning? So are we exactly maintaining the same state? So to study that, like we looked at the time correlation of these states. So here, like when there's no delta, we see that the correlation, the states go to 0. So on y, we have probability to modified at time t, and on x, what we have is probability to be modified at time t0. So a perfect x equals y curve would show good correlation or maximum correlation, and y equals a constant would be 0 correlation. So here, when there's no reader-writer process, the states decay to 0. And with sufficient enough reader-writer term, we are able to maintain the states, but it's a y equals constant situation. So there's no correlation. So we can maintain the states, but we're not exactly maintaining the state that was originally created by the enzymes or by the painter process. So there's no correlation with the previous state. So what does this mean? This basically means that let's say if a modified state is repressive state, like it's a repressive mark, so we can basically maintain the repression but not the exact ChIP-seq profile. So I pose this question, like, can it actually be called epigenetic memory? I pose it to you. So now let's look at how our chromatin region, like how our chromatin state can be translated. So for this, we introduce a cooperative switch-based transcription model. So considering modifications as a form of repressive marks, when there are very low repressive marks, probability to be M is to be modified is very low. We have actively transcribing region. And when in our region, when in our chromatin region, like we have high modifications, we only have leaky transcription activity. And D is a parameter which controls the steepness of the switch. So now let's look at how gene expression depends on the position, like how can we capture variegation with this model. So here I have a similar profile, which similar to the one which we have seen. Like, so there are two profiles here actually, red and blue. The difference is that blue, the process is actually very fast compared to the transcription, and red, it's very slow. So slow and fast epigenetic fluctuations, the fluctuation between M and U state, that's what we're talking about now with respect to transition. So in the case of blue, the process is fast, and in the case of red, the process is slow. But we can have the same steady state profile by keeping this ratio constant. So in our chromatin region, like in our 200 nucleosome region, we have a specific profile, specific ChIP-seq profile for the mark which we are studying. And if we have, let's say we have a gene in between around the position 0. So we see that the amount of marks is high And if it's a repressive mark, we expect the region to be repressed, and we expect low transcription or low or zero transcription from the region. And if the region is, if the gene is here, somewhere here, like around the critical point, like, so the critical point where this transition happens between active and no transcription. So if the gene is around here, it is very sensitive to the epigenetic modifications. So it can either be active or silent. So this would be the case in some of the poised or bivalent genes, like in the case of developmental genes. And very far from the repressed region, like we can have active transcription. So that would be somewhere here. So with this model, we can actually distinguish between the kinetics of epigenetic dynamics. Like we can have different transcripts uh, if the fluctuations or the kinetics of chromatin state is different. And also we can have different gene expression relative to the position of the gene relative to the position of the painter or sequence specific recruitment. Now, with all this, like how can we actually build it up to predict some experimental results? So here we'll consider a study which was done a few years back in engineered yeast system. So basically they were trying to decouple the sequence specific and reader writer effects. So here the system was that they have CLR4 is a methyl transferase. So they have this enzyme tethered to a particular location, and then you have it deposits methylation marks in the gene, and then you have repressive state, and you have a specific phenotype, red phenotype for that. When they add tetracycline, the sequence-specific enzyme unbinds, just like the case of our epigenetic memory assay, where the enzyme unbinds. The enzyme unbinds, and then the other processes like nucleosome turnover, replication, and enzymatic removal of marks remove these methylation marks, and the region becomes active, and we have active phenotype. So this is the system, and they study this system for 4 different mutants. So here, in all the cases, like initially you establish a methylation profile, a repressive state, and then we add tetracycline and the enzyme unbinds, and we modulate, or we basically study the evolution of marks. Or in this particular experiment, they're actually looking at the gene expression, like proteins from the region. So case 1 would be where you only have tethered enzyme, like sequence specific enzyme and normal demethylase. There's no reader writer activity. So in the model, like we don't have any reader writer activity. We have high value of K0, which actually takes the modified state to unmodified. And in case 2, they basically mutate the demethylase. So there's no demethylase, and they have sequence specific enzyme which unbinds. So this is the model equivalent here. So in both these cases, they observe the same change from repressed state to active state. So even without demethylase, all the marks are sort of removed in this timeline. So basically, they're looking at time frame of 100 hours. And in case 2 and 3, they basically add the reader writer process. So the reader writer with active demethylase and the reader writer process without demethylase. So in the case where we have a reader writer process, there is actually a competition. So the reader writer is trying to maintain the chromatin state, while the demethylase and other processes are trying to remove the chromatin state. So this is more like a wild type situation where we have reader writer and demethylase. So in this timeline, like the cells do switch to active phenotype. But when the demethylase is mutated, we do see that like the chromatin state can be maintained for about 100 hours. So now I'll show you how the kinetics work, like how this decay works. So on the Y, we have fraction of off cells, like the fraction of cells that are repressed. And then we are monitoring that over time. So here uh, we establish the repressive state, and at time equals 0, the enzyme unbinds, the sequence-specific enzyme unbinds, and we monitor the amount of, or the fraction of cells that are still repressed. So case 1, where we only have the sequence-specific enzyme, at time 0, the enzyme unbinds, and very quickly, like in about 6 hours, the all the cells turn active, and we have the active phenotype. Just to remind you, so the scatter points are from the experiment and the lines are from the simulation. In the case of blue, where we have sequence-specific enzyme, but demethylase is mutated. So even when demethylase is mutated, we turn these cells, like the fraction of off cells, decay to 0 in about 20 hours, and all the cells turn active. In the reds case, the difference is that we have reader-writer process. So the reader-writer process is actually trying to maintain the cells in off state, but all the other processes, the enzymatic removal of marks and replication and other processes are trying to take it to the active state. So there's a competition, but eventually what happens is that like all the cells turn active. In the magenta case, we have mutated demethylase. So when the demethylase is mutated, in this scenario, the reader-writer is almost able to maintain the states, like a significant fraction of the cells still remain repressed in about 100 hours time. So 100 hours is actually a significant time in the life cycle of yeast. Just to recap, so if you compare between blue and red, the only difference is that we have the reader-writer process. So when we have the reader-writer process, the decay is much slower. And if you compare between the blue and magenta, uh, still the difference is that you have the reader-writer process. So um, in the case of magenta, where we are able to maintain the states for much longer, with demethylase mutated. So in summary, we can actually capture the kinetics of this decay, like in all 4 cases, very well with our model. To conclude, so now we have a generic and simple mathematical framework for the chromatin state dynamics with some mechanistic insights. Also, we can relate the model with the experiments, and we can predict experimental observations. With that, I'd like to thank you all for your attention, and our group in Lyon, with Daniel and Cédric. Also, Sarah and Lynn, who did the animation and the organization of this presentation. Once again, thank you all for your attention.